into the rest of the class, this material and the principles you've learned here will sort of feed into that next material. And finally, the third one. It's not like a comprehensive final in the way that you have to go back and revisit all the stuff that. So there's that. Leia, come on, you're causing a distraction. Sit. Yeah, sit. Sit, please. All right. The other reason is because I am always amazed that most of the students in the class have had very few experiences with short answer exams. And it tends to take people a little bit by surprise. And so I downweight the first exam and gradually upweight them to account for that.
And these are curves?
They're all curves. Usually that amounts to maybe it's 80 and above that ends up being about an A, generally. What I do is, after we have the scores, then I tell you this is your score and this is your letter grade equivalent for that score. But I curve the whole course, actually. So this letter grade equivalent is not like a fixed thing. It's just to give you some sense of where that's falling. And then ultimately I do the same thing with your accumulated scores at the end of the course. The grade distribution in here tends to be similar to other grade distributions in other biology classes at the end. You guys would be raring to go. Go ahead. Start up.
I'm going over like the clock, like a monthly clock or whatever. I don't really get like the whole.
Okay, so the principle there is,
You say decide.
Yes, that makes sense. Actually, we may pull down the thing and pull back up the Clifford question. So the principle there is that, let's throw off phylogeny. The principle is that if we were to look at a particular gene, we have those Clifford questions that refer to this fictional gene, Leia. That's a beat. That refer to this fictional gene, HYR. And the idea is that over the course of this evolutionary history of a group, that this gene is mutating and those mutations are accumulating at a constant rate. So that means that on average, when we look at each of these branches, what we see is sort of a pretty constant rate. I'm going to draw it very evenly so that I get a constant rate, but it would not occur evenly. We're seeing changes accumulated. Okay? So then the principle is, if we compare any two members of this tree, we should be able to recalculate that rate, and we should be able to apply it to the rest of the tree. So just as an example, 1, 2, 3, 4, 5, 6, 7, 8. There are 8 changes here between these taxa, C and D. And if I tell you that this is 8 million years, then what you could
from the ancestor to
exactly. Then what you could say is that the changes are accumulating at about 1 per 16 million years. Am I doing the math right? 1, 2, 3, 4. 4 changes. Oh, yes. No, I'm sorry. 4 changes per 8 million years. on average, which would make these guys differ by 8 changes. That's
Why would
1/2
they
of the million.
differ by 8 changes?
Because the changes, the 4 per 8 million years, are accumulating along each branch. So there aren't just 4 changes that distinguish C and D, there are 8, because they have accumulated on both branches. Right. And then if I asked about, well, how does B differ from C versus how does B differ from D in the number of accumulated changes or the percentage change, those should be equivalent, right? Because this is just another branch that separates them, and it's exactly the same amount of evolutionary time dividing it.
So 8 million for
The 8 million, I had to tell you. Yeah. Without that, I know that my relative clock holds. Okay, so I expect on average the same number of changes between here and here, and the same number of changes between here and here, and all that other stuff holds. There's the same expected number of changes between B and C and B and D. So if I told you there were 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. 12 changes accumulated between B and C, and asked you how many changes should have accumulated between B and D, your guess would be 12. Because you expect there to have been the same amount of evolutionary time, the same amount of change accumulated.
What does that have to do with the 8?
8 is real time. So you can put real time on this, and now I can say more. I can date this change, for instance, as being about 50% older than this change. So I would say this happened at, and I'm doing my math wrong, 12 million years.
And that being because there's 12 instead of 8.
Right. 2 additional changes, about half as many as accumulated in 8 million years. So that adds another 4 million years. That's pretty fun. That's pretty fun. Did I cover it enough?
So if you went back between A and B, and there were 16 million, or 16 changes between A and B, would it put that to be
16 million years.
years?
Yeah. Well, between A and B, how many changes between whom?
16 between A and B, because you had 12 between B and C.
Yeah, 14.
When I put this 12 on there, I just looked at where this node fell relative to a single line.
But there are still 12 changes between B and C.
Yeah.
Is
So,
that equal to 12 million and 12 changes?
well, in this case, yes,
The 8.
because, all right, so this is where that doubling thing is happening. And I should have used odd numbers to make it more obvious. All right. On this one trajectory, let's just look at this single line of evolution. What we know is that 4 changes accumulated in 8 million years. And that back to this node are 2 additional changes.
Okay, so that's an additional 4 million years.
Right. And that's why I wrote down this 12. Now, because 4 changes occurred in 8 million years, and when we compare 2 daughters from that ancestor, we double that number because it's accumulating on both branches.
So you expect another 4 million to happen.
There's another 8 million worth in terms of time of change.
I mean 4 changes.
Another 4 changes have accumulated, yes. And so, from down here, yes, I have 6 changes on this branch, and I do expect 6 changes on that branch. But the rate of accumulation per year is happening in either branch, so I'd only look at one branch
So you'd
to
expect
say what the rate is.
if there was another 2 million years between, or another 2 changes between
A and B.
and B, then you would expect it to be 16 million.
Right.
Okay.
see that there's some error here, right? I guess if I was drawing it religiously, it would be there, and I would have 2. Yeah.
So then you would expect A to have 16 changes, and, or no, A
Eight.
to have 8 changes, and then B to have 8 changes. Okay.
Which is what we see. Yeah. Now, it is honestly true that this is on average. So it is possible that these branches don't have precisely the number of changes. And sometimes they differ so much from expectation that you'd be tempted to say that there's something more going on a particular branch than simply the clock. And so that would be one means by which you might evaluate whether a particular lineage was evolving at a faster or slower rate than you expected. This is what you would expect. I think you were ready to ask a question, no?
I just want to know, like, how accurate is this, like, method of calibration?
It's really interesting. As you try and extrapolate far from where you've actually measured the clock, your error gets bigger and bigger, just as any extrapolation. Some loci tend to have quite clock-like behavior, and others, messier. So the sorts of things that you would want to say, if you wanted to extrapolate way back with the clock, you would attempt to validate the clock within the time frame that you have as strongly as you can. By saying, okay, when I look at these recent branches, which I have since my calibration, they all behave very nicely. It suggests it's a very regular clock, and then you'd go and extrapolate. But you'd have big error bars.
Yeah. So the farther back you went on this, it would be more error.
If my date is 8 million, this is the date that I have. All these other dates we're talking about, we're calculating. So as I go further and further away from my 8 million information, yeah, I'm expecting more and more error. Yeah, go ahead.
Okay, when we're trying to determine if there's anything else going on besides just the changes, would we be thinking about the heart behind the like migration, drift, like
what has caused these differences? Yeah, you would. And in particular, you would expect there to have been probably selection at this scale. In part because once you are at the point of a phylogeny, fine scale migration amongst populations has all gotten washed into that, right? So what's there in the clock is the drift expectation. And as you deviate from that, you would interpret that as some form of selection.
So these wouldn't, I was thinking that these would be mutations.
These are mutations, but they're only accumulating via a drift process. On average, right, we're thinking that the majority of mutations are neutral, the ones that accumulate. Occasionally there's some beneficial mutations. So for instance, if you saw on one of these branches that you have more changes than you expect, you might interpret that as positive selection, or selection for change in this particular locus along that branch.
so if you would see more changes in a branch?
Yeah. I think that's a reasonable interpretation. Now, another possibility is that you have selection or some source of variation in mutation rate that's very strong. So again, if you saw that in a particular branch there was much higher change, one possibility would be that you had higher mutation rate in that lineage.
Can you go over, like, calculating the rate of mutation?
The rate of mutation. Good. I'm not sure.
Like,
So, what we figured out with our calculations. Can I still be here, or should I?
Yeah, that's
sure exactly what more you want. I'm not going to ask you to recapitulate that, just to know it.
So, they're in proportion to the rate at which they occur the rate of.
That's right, the expected rate of fixation. So, let's put it this way. If the rate of mutation is mu, maybe you are getting confused about this issue of how many of these mutations are neutral versus not. But we expect that the majority of them are neutral, have no effect at all, or nearly neutral. And so some large fraction of them will drift only, basically. And so the rate of fixation of new mutations is mu. You could also be thinking about what we said about a given mutation. So what's the probability of fixation of a single mutation that we are interested in? We've seen this mutation occur. What's the probability of its fixation? That's I think this should come on. The probability of fixation of a particular mutation via drift is simply its initial allele frequency, right?
The probability of mutation of one.
Of a particular new mutation.
Okay.
It's just its allele frequency, because the probability of fixation of any allele is its allele frequency. And so if it's a new mutation, at a particular locus, you would say its probability of fixation is 1 over 2n. It's just one allele out of all the population of alleles of that locus. And then we went on to say that if we think about the number of new mutations like this that could arise, we recognize that it could arise in any of the alleles. And so the number we expect is mu times 2n. And that's where these crossed out to give us our expected rate of fixation of new mutations. This is what we were thinking about.
Yeah.
Mm-hmm. Go ahead.
And then, can we go ahead and do number 3 on the practice exam? It's very interesting.
All right. You'll have to tell me what it is. Hold on a second. Let me erase it.
How similar is our examination to the practice exam?
The practice exam is made up of questions from former exams. So the style of the exam is similar. Oh, thank you so much. The style of the exam is very similar. The particular scope, the particular questions I ask varies a little bit from semester to semester. We end up spending more or less time in part based on questions that arise in class, and I feel like I like to have the exam be representative of our classroom. Okay, so number 3. This one gives you a table of 5 populations. And they each have their effective population size.
he's provided, except for RD1 words.
Those basic allele frequency calculations, yeah, those you need to know on your own. And then it gives an initial P. And it asks, part A is give the probability of fixation of this allele A1 and the estimated time to fixation, assuming only genetic drift is acting on, for each of the pots below. Provide a ranked order of probability of fixation and time to fixation if you cannot provide the estimate itself. That's part A. And then it asks you to define effective population size and explain the effect of NE on genetic drift and why it occurs. Okay, let's do the first part first. The probability of fixation. Is equal to the initial frequency. So you would just copy these numbers into this list. Let's do this. Okay. The time to fixation.
And that would also be the probability.
Well, these alleles have already accumulated a little bit. So what's happening with these alleles is the same
thing that happens with mutation. It's just that mutation arises for the first time. Yeah. And then the time to fixation that it asks you, we do not know the precise time to fixation, but the time to fixation is inversely related to the population size. So you would say that you're giving the rank, and this 1 is soon, 5 is late, so we have 5, 4, 3, 2, 1.
So you're solely basing it off of the population and not discarding
Right.
the frequencies.
Right. Because it is the population size that results in those little incremental changes.
So even though I think we did. No. So even though the 1,000, the initial P is at 0.8, it's much more probable to fix before, or
Okay, so remember, some of these are down here. One of them is up around there. I'm just doing them arbitrarily. The other one is up here. And 0.3. Most of them are down here. But 0.8 is just as close to a fixation point as 0.2. So you could suggest that this 0.45, it's the farthest from the potential of fixation of one or the other allele. So perhaps it would have to drift longer. It also has intermediate population size.
But just for this question, it would be
For this question, you would go with population size. Yeah. You would not worry about the subtlety of initial allele frequency. Okay. Part 2 of the question, which we would carefully label, make our grading easy.
ask you about what is effective population size. And basically this is the population size, the idealized population size. That has the same genetic properties, basically, or how to put this more distinctly.
Isn't that where they take the men, or the amount of males and the amount of females?
Mm-hmm. This is the idealized population size where everyone contributes to reproduction equally. That has that behaves the same. As my focal population does genetically. Okay, so it's the idea that you have a population you're interested in, and if you could create an idealized population that behaved in the same manner genetically, but had everyone contributing to reproduction equally, how big would it be? And that's what your effective population size is. The second part of Part B asks you to explain the effect of NE on genetic drift and why that occurs. So what you would say in this question is that it's the effective population size, not the census population size, that describes how this population will change over time. And so even if you can count 10,000 individuals in your population, if your effective population is more like 2,000, then drift will be acting more rapidly, be a stronger effect, because you have a smaller effective population size. So in other words, you'd be explaining why would we bother to use effective population size. What was it? The effective population size of whale sharks.
How many of you read
That
that?
was like 100 and something, 200.
Oh, wow. Yeah, really low. Interesting. Okay? Thank you. You stay. You're not going anywhere. Mm-hmm. So we only mentioned it, really, so far. So keep that in mind. We will come back to it in this next portion of the class. But we've only mentioned it so far. And what it describes is that it's possible for selection to stem from the frequency of alleles in the population, or as we'll see when we discuss it in more detail, the frequency of phenotypes within a population. And we talked about this scenario of males of different types. One strategy works better in the presence of the other strategy than it works as its own strategy increases in frequency. This is the sort of thing where you would have to be given quite a bit of information to do any calculations. You would have to be given some relationship between frequency and fitness so that you could do some calculations with it. I will emphasize again that we have only mentioned it. The reason we mentioned it was because it's one of very few mechanisms to explain intermediate equilibrium allele frequency. Most of the selection that we've talked about, we expect to result in fixation of one or the other allele, or one allele over time. Drift ultimately results in fixation also. So we only have very few explanations for why we should see multiple alleles maintained in a single population. Heterozygote advantage is the one that we've talked about in detail. Frequency-dependent selection was the second one. Yes, that's why I bought that for you.
Was that the one like where if you had bees and there was one plant that had more pollination than the other, and they would give another plant to pollinate that more? So like the frequency of the plant.
Is this an example I did? Or did you read it in the book? I think the book probably talks about this. Yeah.
So it's that.
It's that scenario.
Yeah. Okay.
Mm-hmm. Yeah.
How did you get in?
I talked about male. Yes, definitely. Do you guys have some recollection of heterozygote advantage? You know what it is.
Heterozygote advantage.
Yeah.
Basically, keep the second allele alive, no matter using it to double the sensitive length, it's still staying alive because it's heterozygote.
Exactly.
So the frequency of dance selection is the second reason why the recessive allele will stay alive, even if it gets selected out.
Right. Yeah.
So even if it's not necessarily saying that in heterozygote form there's more fitness.
No, in heterozygote advantage, for sure that's true. Yeah. In frequency-dependent selection, the fitness depends not just on the combination of alleles, but the frequency of those alleles in the population. Yeah. And I'll just mention that heterozygote advantage leads to a stable equilibrium frequency. Remember we talked about this difference between a stable equilibrium frequency where you can change the allele frequency away from that allele frequency and it will come back to it. That's the stable equilibrium. And this differs from the Hardy-Weinberg equilibrium because as you remember, as soon as you change allele frequency, however you do it, with migration or with random effects, you get a new Hardy-Weinberg equilibrium that is associated with that new allele frequency. And so that's a neutrally stable equilibrium.
The heterozygote advantage is like the model of the wall, right?
That's right.
I always think about like S is how many will not survive, right?
Right. So the fitness, the relative fitness of any genotype that has lower fitness than the maximum is 1 minus S. And when we're talking about heterozygote advantage, remember we had 1 minus S and 1 minus T. Yeah. That was just to recognize that the equation, the fitnesses may not be, oh yeah, that's right, I'm over here, may not be equivalent of the two homozygotes in a heterozygote advantage case. So for instance, these are our relative fitnesses. In a case like this, where we have heterozygote advantage, we know that the heterozygote, its relative fitness is 1. because that's what heterozygote advantage dictates. But then our homozygotes can differ from 1 in different amounts. So, this is 1 minus F, this is 1 minus T. Just to recognize that it doesn't have to be symmetrical. If it is symmetrical, what's our expected genotype frequency over time? Our equilibrium genotype frequency, allele frequency, excuse me. Yeah. So if it's symmetrical, then the equilibrium allele frequency is 0.5, which means that the equilibrium genotype frequency is 0.5. Yeah. And of course, if it's not symmetrical, you still expect an intermediate. But these T and F values will tell you immediately sort of where should it fall. So if T is bigger than F, then the allele frequency should be dominated by the little a alleles, right? It should be, in terms of P, the allele frequency of big A, it should be greater than 0.5. All right? So this is
they're both selection coefficients.
They're both selection coefficients. We're just recognizing that they can take different values. Yeah.
You're saying if T is bigger than F, then it's dominated by a little, right?
Yeah, then our population will be common. Did I say it's backwards? You're right. I said it backwards. Then, if we think of P for big A and Q, let me just write it down so we get it straight. Because you're right, I said it backwards.
Okay.
So if T is greater than F, we expect PA to be less than 0.5 and QA to be greater than 0.5. And if F is greater than T, then we expect P to be greater than 0.5 and Q to be less than 0.5. And of course, we've just gone through the case where where S and T are equal to one another. And that's this case where P will be equal to 0.5. You'd be able to say, when you have S and T, actually, you can calculate this. And I think, I hesitate to do this because I might be getting my math wrong. But I think
She's like 100.
it should be like this. Am I getting it right? If T is greater. Let me just check. 3 is greater than this should be less than, no. So then it should be S over T plus F.
But that's always the case?
Yes. Check your lecture notes and make sure I have not given you the wrong value.
T is T over F.
It is. Okay. So then
Is it the total?
Oh, yeah. No, this isn't making sense to me.
Yeah.
That's right.
Well, because the way that you did it in class, your AA is 1 minus F, and then your little a is 1 minus 2.
Okay. Okay. So, what that means is that according to this example, it will be F. Let's be clear about what's going on here. This formula, the value of P, is the selection acting on the other allele over the sum of selections acting on the two alleles.
Okay.
Yeah.
So the selection acting on Q allele.
Mm-hmm. That's the key. Thank you. I'm glad we ironed that out. Mhm.
So will you give us formula on the exam?
Yeah, that would be a formula you'd see on the exam.
Okay, you would specify that, like, say P is the selection coefficient for big A, and then S is the.
Actually, it would just appear sort of like that, and you'd have to think about it
Okay.
and be sure that you knew what you were doing.
And because depending on like the way that you wrote it here or the
Yeah.
way that you wrote it in class, there would be different formulas.
Exactly. I will probably be religious about doing it the way that was done in lecture.
Okay, so
So what you would see probably is P is T over S plus T, but you would probably be calculating in this case what S and T were. So you would. I think there's one on that practice exam.
Yeah, it's
Which one is it?
I think it's number 1.
This is the one with hamsters?
Yes.
Yeah. Let me take a look at that. We'll do that one.
yeah, I remember talking about that with you
Yeah.
after class, but we didn't actually do it.
All right. So in case you haven't read this problem, this is the idea. This is a hamster problem. And it gives you the true fact, which I think is so interesting, which is that the entire pet trade of hamsters has resulted from a parent population of 4 hamsters, collected from the wild. Yes, that is actually true. And so the diversity that you now see has all been bred from mutation. As it turns out, this is what it says, with different strains of hamsters. For instance, the black bear hamster, this is the short-haired, black with white feet, very cute. And the Dalmatian hamster, this is short-haired, white with black spots, also cute, but not as cute as the black bear. There are often fitness
That's not
advantages.
scientific, huh?
No, that's associated with increased heterozygosity. And in one example, crossed hamsters lived 3 years. We're going to call these B. Crossed hamsters lived 3 years. These are the absolute fitnesses. Think of them that way. While the black bear hamsters lived 2.4 years, the Dalmatians only lived 2 years. What equilibrium would you expect in a freely breeding set of hamsters? I think I have all the information I need from this. Thank you. All right, so our first task, because we have absolute fitnesses, is to calculate relative fitnesses.
And by you mean that that's the fitness that applies to all of them, not just the relative fitness of each one.
Right. You're confusing me with that statement. All of whom?
Like each one is affected by this one fitness.
So I would interpret the question as the black bear hamsters live on average 2.4 years. And so it refers to the whole population of that genotype.
Not just
a particular one that I have in my home. Yes, think of it as the average. So the relative fitnesses, to calculate relative fitnesses from absolute fitnesses, do you have dogs? No? Usually only approaches people that have dogs. Is to take the proportion of the maximum fitness from the observed fitness. So of course this is going to be equal to 1. There. Moving instructions. Come on. 2/3. 2.4 over 3. Anybody have their phone do these calculations. This is 6-7, of course, but
This one is 0.8.
thank you. So now we have our relative fitnesses. And we'd be ready to apply this formula. But they're not expressed in terms of T or S, right? So using the formula as we did in lecture, let's calculate this as 1 minus S. And so S in this case is 0.2. Over here, 1 minus T. Point 34. 3. Oh, yes, you're right. And now I apply the formula. .33 over 0.53. What do I get for that? 0.6 something. So in a freely breeding population of bear and
We're assuming that
Dalmatian.
DB is, or D is the P allele.
Yeah, in the way that I've done these calculations, yeah. You could have calculated the opposite way. You just need to keep track of which goes with what. So that's one of the reasons why it's useful to remember these patterns. You should know approximately where those numbers are going to fall based on the selection pattern. So given that there's higher relative fitness of this DB, the bear, as opposed to the Dalmatian, I expect to find more bear genes than Dalmatian genes in the end. I can start off by saying that. So if I did this calculation and I found that this value was on the other side, I would realize, oh, I've got it reversed. It must be the opposite of that. So that's how you would do that one.
Really the second part of that question, or what was the final question of that?
I think this is the final part. This is the equilibrium peak. I think that's the only part of the question that I saw.
Yeah.
Yeah.
That's for B, right?
For B.
Subtract from 1 gets B.
Yep.
Mm-hmm.
I think heterozygote is advantage is fair game, there's no doubt. We spent a lot of time on it. We did it in lab, we did it in lecture. Yeah.
So when you do the test exam, is that when we take tests for the
It's
actual
7 questions.
It's only
8,
8.
yeah. The exams routinely are 7 to 8 questions. As you notice in the practice exam, each question has multiple parts. And they're usually broken out by A and B or A, B, and C, or I try to break them out explicitly. So there are multiple questions in there, even though there's only 7 or 8 total. And we're breaking out credit to all of those things.
I'm assuming you do partial credit.
Yeah. Oh, yeah. I think I can venture to say this, that it is true that on most of my exams, very few people get full credit on most of the questions. Usually the high on the question is a little bit less than the total point. Partly that's because I think we set our total points with high expectations to recognize those of you who really get something. And then, because we're curving, okay, nobody happened to pull out a piece of it, that's okay. We just rescale. But it highlights, again, do what you know. You're working on a question and you get to a section of it, part B, it's like, oh, I don't know this part. Go on to part B. Or move on to the next question, come back to it. Don't spend a lot of time trying to figure out stuff that is not coming to you easily. So polygeny is the general term.
Oh, it's quantitative and then it's on it?
No. But the previous lecture, the Monday lecture of this last week, that is on it. So polygeny and epistasis and linkage disequilibrium are all part of the exam, but not quantitative genetics, which was just Wednesday's lecture. So polygeny just refers to the fact that there are multiple loci that contribute to a particular phenotype. Those loci, once you have polygeny, those loci could have epistatic effects. Well, okay, we can say more than that. We can say once you have polygeny, you can have epistatic effects. I'll just say this. Say again? That's right. If it would be epistatic if there's some interaction between the genes. If another way of saying that is if there's multiple locus dominance, you can think of it as multiple locus dominance, or fundamentally any time that the phenotype observed for a particular genotype deviates from what you predict based on the action of each allele within its locus separately. Okay? So there's epistatic effects or not, and this is the case where just as he said, the expectation of phenotype matches the predictions from the independent action of the loci. Not epistasis.
What are these predictions like? I don't understand what you can predict to see if the deviation.
So you remember where we had those clicker questions about this color?
The flowers.
Yeah. So that was a good example. I'm gonna bring it up.
We don't have to know, like, what the ratios are.
The importance of the different ones. What do you mean?
Yeah, what's the ratios for?
Oh, the Hardy-Weinberg multivariate thing? No, I won't expect you to do that without formulae. I was wondering whether, as I was walking into the building, whether I had remembered to post this lecture up or not. But if it is not there, I will post it directly after because I'm going to check. Can't remember if we did that Monday. Maybe so. Let's open this one first. Monday? Yeah. That's what I was thinking too. Alright. It's okay, Leia. I know it. It's okay. You're fine. You're fine.
I think it was February 8th, whatever day that was.
That doesn't help me.
February 8th, and that's Wednesday.
Wednesday, okay. I think that's the one I'm pulling up. Oh yeah, here they are. Fantastic. Okay, it's going to come up onto the screen in a minute. You're okay. Come here.
February 8th. I don't think I can move my left hand.
No. Just stay. Come here,
February 8th.
Come here, you. You're being ridiculous. Come this way. It's all right. Okay, you stay with me now. Crazy dog. Okay. Here it is. I can make it a little bigger. So the idea here was we're sharing a relationship between phenotype, in this case flower color, and genotype at these two loci C and S. And obviously I haven't fixed this version. Suggest I haven't posted it. And your task then is to ask, can I describe easily the independent effects of each locus? And if you cannot, then you would say, oh, there must be epistasis. So in this case, you can see that the C allele is controlling color, because across these C allele presentations, we have sort of red and blue, or purple, and we have blue. In the S case, the big S appears to be controlling the brightness, and it appears dominant to the little s, because we see that there's bright coloration in all of these individuals that have the big S allele, whether it's 2 copies or 1. And then we have a paler formulation of the color in the little s. And they all pretty nicely add up, right? You can easily see what's going on with the C allele, and you can see that sort of what's going on with S allele is added on to that. Using an example one of the students in class made on Wednesday, if, for instance, these little s individuals, no matter what was going on at C, all had pale pink flowers. So you saw this pattern with these other sets of alleles, but then there's pale pink across the board here. That would represent a case of epistasis. Because your expectation would be that as the C locus changed, you should be seeing the color change from red to purple to blue, and you would no longer be seeing it. And that would represent an epistatic interaction. Okay? So this is the degree to which you'd be asked to look at a system and interpret whether there was epistasis or not. Yeah, I think that's the sort of level of yes and no that we would ask. Because once you have epistasis, the challenge to explain how the two loci interact, this requires a lot more data. Let's say we had this case where the little s individuals all had pale pink flowers. I might ask in this question, is there epistasis and how do you know? And you would say, well, because we no longer see the action of the C allele, which appears to control color, when the little s allele is present. Or there appears to be dominance of the little s allele over the C allele. Either of those would be a reasonable expression. So I'd just be asking you to explain to me why you think it's epistasis and not just simple polygyny. Okay?
Can you go over the difference between synonymous and
non-synonymous?
well, I know not synonymous. What can you just go over that, and then pseudogenes?
Oh yeah. So pseudogenes are just copies, accidental copies of existing genes. And initially they have really no function whatsoever. They don't. Nothing is being transcribed around them. So that they have the potential to accumulate even more mutations than do coding regions that have silent mutations.
Because they're not being paid attention to, essentially.
Yeah, right.
And then they can go on to create.
They can evolve over time to have new functions. Yeah. Which is a process we know happens regularly, because we can see gene families and we can see this process, but we don't know all that much about how it happens. How does a region of DNA that has had no function whatsoever go on to evolve a new function and regulation and the whole deal? This is an area of research. We don't know that much about it.
Okay.
The case of synonymous substitutions are just those substitutions that don't result in an amino acid change. So
like a T.
So the third position in a codon, the majority of times, does not result in a change in amino acid.
And that would be a synonymous.
That would be synonymous.
But if it did change it, it would be not.
Right. Right. And we discussed in class that although we think of synonymous substitutions as being neutral, and effectively they are neutral in most contexts, it's possible for them to be selected against if the change, because they still would produce a different sort of mRNA, or rather tRNA, as that transcription process is going on. And if those particular nucleotides aren't present or in different frequency, there could be slight selection on it. That's what Arthur was talking about in lecture that day, when I couldn't remember what the selection was caused by. So because transcription is still going on, and those third codons are transcribed, it's possible for there to be subtle differences in the speed at which they can be transcribed, even though they ultimately code for the same amino acid.
So a neutral mutation can be either synonymous or it can be a mutation that changes the amino acid but doesn't make a difference in the phenotype.
Exactly.
Okay.
So there's plenty of parts of proteins where a change has little or no effect on the functional portion of the protein. Because remember, proteins, the vast majority, they're all folded up, and then they present, one or a small number of active sites. And as long as the active site is still functional, still working as it always did, the rest of the change really doesn't matter. It's entirely neutral. I would just look them over. I think one of the things about the clicker questions is they're just sort of checking how you followed, what went on in lecture, and what you remember from lecture. And in all of these cases, in part because they are clicker questions, I more or less expect you to be able to approach the problem. It might take you a moment to reason through it, but it should immediately be obvious what to do with each of these clicker questions. And so I think that's a good guide to, okay, my base knowledge should be this.
Can we just go over
linkage disequilibrium? So linkage disequilibrium I'm going to put up this question simply because you would be given a formula if you were asked to make some calculations about this. Linkage disequilibrium represents any case where the frequencies of particular gamete types are not what you would predict simply based on the allele frequencies in the population. So, yeah, that's okay. It refers to any case where the allele frequencies in the population are not what you'd predict based on, or excuse me, the gamete frequencies in the population are not what you predict based on the allele frequencies in the population at two different loci.
So basically, definitely not in Hardy.
This is another measure of not in Hardy-Weinberg, yeah, that stems from, so it's possible that each of those loci would be in Hardy-Weinberg, but their combinations are not.
Okay.
So it is sort of a form of deviation from Hardy-Weinberg, and the book talks about it like that. So the sources of this kind of variation include selection, and that could be both positive selection where, for instance, some combination of alleles is favored over other combinations, or selection where particular combinations of alleles are being selected against. It could stem from non-random mating. We'll see in sexual selection we have the formation of linkage disequilibrium because individuals with mating preferences tend to mate with individuals that have the things they prefer. So non-random mating will generate this. Actual physical linkage will also generate this, right? If these two loci are close together, then they cannot fully assort independently because recombination is not occurring freely between them. So this value D is calculating the difference between the product of two types of gametes and the product of the other two types of gametes. And if we think of A as having, I'll write this out, let's see. You're good, you can stay there. You want me to move that? If we think of A as having P and Q allele, there you go. I have big A's and little A's, right? Yeah. We're reusing some variables here. Sorry about that. If we think of it this way, then the probability of the AB gamete is basically P times S. Probability of the A little b is P times P, or did I do it right? Yeah. The probability of little a big B is 2 times F. Thank you. It's P times P. So if there's no linkage disequilibrium, then my formula D simplifies to PS
If there's no linkage disequilibrium, D should be 0, right?
That's right, it should be 0, and I'm showing you why it should be 0. Because, in effect, if you combine the sets of gamete types, what you really are putting together is P times S times Q times T minus P times T times Q times S, which you know should be the same thing. And so the equation itself is posing the question, are the gamete frequencies in the same proportion, the gamete frequencies, what you'd expect, just based on their relative frequencies of alleles, P and Q and S and T, explicitly. And so your measure of linkage disequilibrium is the deviation from this truth, this expectation. And you remember it's gonna range from 0.25 to minus 0.25. The directionality is just telling you about which particular gamete combinations are over or underrepresented. Because in essence, it's just looking at these deviations from these expectations. So to calculate this, we went through and we looked at expected values and we looked at observed values. These are your expected values. Your observed values are whatever you observed.
And those would what you would fit in your equation for D?
In your equation for D, you would use your observed values. And that would tell you how you've differed from the expectation. Because you know that with the expected values, you'd get 0. Because they're defined that way. The importance of linkage disequilibrium is really that when you have linkage disequilibrium, these two loci are not evolving independently. No matter how you ended up with it in the first place, they no longer evolving independently because particular allele combinations are more or less likely than you'd expect. For instance, let's say if AB is in high frequency, the majority of A's we find with big B, then that combination of alleles is evolving together. So it's acting as if it's not two entirely separate loci.
So in this question, the expected frequencies are really like that's just extra information.
Yeah. I haven't bothered to give you what these P and Q are. I just told you what these totals are. Yeah. So, you could be given P, Q, S, and T, and you could generate these expected.
Which you would need if you had to do chi-squared.
No, you wouldn't. I mean, you won't have to do chi-squared. We haven't done it in class, so I don't expect you to be able to do it on the test. I do expect you to appreciate that we would ultimately use it, but we haven't focused there. But, for instance, if I hadn't given you these gamete frequencies, I'd only given you the P's and Q's and S and T, and you'd have to calculate these expectations. If you wanted to, for instance, identify which combination of alleles was, yeah, or the greatest away.
Okay, but essentially those values aren't important when it's calculating.
Right. With D, you want to use the observed. Exactly. You could, for instance, the previous question just asked if it was in Hardy-Weinberg equilibrium, yes or no. And you could glance at those numbers and decide without doing the calculation, since I gave you expected and observed. And I would already think, oh, well, one of them is twice what I expect. That's probably not in Hardy-Weinberg. That's probably not in equilibrium. It's probably in linkage disequilibrium. So I would have said yes without doing any calculations for this question. But then in the next question, I asked what is the value of D, and there you would have to do a calculation.
that where you use your range, 2.25?
0.25, that's right. Once you make this calculation, you're going to decide whether you think it's close enough to 0 that you'd say, that's a little bit of random variation, or it's far enough away that this is something going on. And it is valuable in that context, in making that judgment, to ask, okay, what is the maximum value it could have? And so, for instance, we had the example in class where we calculated a D of 0.1. And 0.1 really sounds like a small number, right? That's not very different from 0. But when the maximum value is 0.25, then 0.1 seems like an important difference. Yeah, so that's why keeping in mind the scale of these values is useful.
And how close would you consider to not being in if we're not using it correctly?
Oh, I think if it were less than 0.05, I would start to be like, I wouldn't give you a value that was that close where you have to make that kind of judgment call. But, if you felt like you were making that judgment call, what you would say in your answer would be, I got 0.04, and I judged this to be not different from 0, therefore there's no linkage disequilibrium, which tells me that you know what the criterion is, even if I would have judged it differently. Yeah. But it is not my goal to have you be making subtle judgment calls.
Site-specific recombination would be an example of linkage, a source of linkage disequilibrium?
So usually recombination is going to tend to degrade linkage disequilibrium. In part because it's breaking up combinations that tend to be overrepresented. So we think of recombination as a force that erodes linkage disequilibrium rather than creating it. I'm trying to imagine if there is an example. I can't think of an example, although this would be the sort of thing that one should ponder in a little bit more detail, of whether there could be some sort of restricted pattern of recombination, a form of mutation almost, that tended to combine particular alleles. I can't think of one, but...
Like if you have transpose on the base fission of a gene, and then you bring two genes that were like originally very far apart,
much closer together.
More linkage.
Yeah. Yeah, that would be a case where you've created a sort of all-new ballgame. And suddenly you've got linkage. From that point on, whatever recombination occurred would tend to erode it. But yes, that kind of mutational event could create linkage. Yeah. Are we good? It's not going to be bad. It's only worth 15% of your grade.
Very intimidating.
Yeah, I'm worried.
I know.
Yeah, I'm like, if you flunk this, you can't get an A.
I don't think that's true.
Yeah.
Yeah, exactly. This is a really good way to think about it, which is that in the grand scheme of 100 points in the course overall, this is 15 points. And like you said, if you got half the credit, you would still have 7 points out of 15.
It doesn't give you very much room for error, I'll put it on.
Well, see, the curving is impressive in this class. See, I've been avoiding that question. The average is usually probably between 55 and 65.
Yeah, but I think you still said this.
Okay, good. I've been honest about it.
So, but a 55 would be essentially like...
That would be like getting a 75 on a normal test.
Okay. Yeah, that's pretty average. I mean, that's pretty normal, though. Like,
That's not what students tell me.
Well, when you apply the curve.
When you apply the curve, it's all normal. When you apply the curve, it's all normal. There's no doubt about it. But then if you focus on the numbers, you'll feel stressed. Focus on your equivalent letter grade. Good luck to you all. And I apologize again for oversleeping. That was just unbelievable.